Hi there. In this video, we're going to be continuing our introduction to neurons. And remember last time we did a basic review of cellular physiology, properties of the membrane, and basic ideas in ionic chemistry. Again, if any of these are unfamiliar to you, go back, check out these sections of the book. These are very important concepts you need to know. And today we're going to actually be talking about neurons themselves. We're going to be talking about their basic structure. We're going to be talking about different kinds of neurons. And we're also going to be talking about a different class of cells in the brain called glia, which are not neurons. They don't perform the same function as neurons, but they do have very important supporting functions for maintaining brain function. Now, to do this, I want to start off with just a sort of a little bit of neuroscience history. So I want you to imagine that you were a scientist, a biologist, working back in the 1800s. And in the 1800s, you would probably know about microscopes, and you would know that you could take tissue from living things, and you could examine it under a microscope, and you could see that living tissue is made up of cells, and you would know that there's something important about the fact that living things are made of cells, and that cells do important things. And if you decided to say to yourself, well, I want to study brain tissue. I want to know what brain tissue looks like when I put it under one of the microscopes that I have available to me. And if you did that, you would see something like this. I mean, maybe it wouldn't exactly like this, but it would look like just a whole bunch of indistinct mush. You might be able to make out some blood vessels or a few other little features here, but what you'd find is that you really wouldn't be able to see any distinguishing features in brain tissue, in unstained brain tissue under a microscope. Now, all of this changed in the late 1800s when a scientist named Camillo Golgi created a new kind of stain for neural tissue. Remember, we've talked about how you can impregnate neural tissue with a stain that labels cell bodies, or a stain that labels axons. Well, one of the first stains that was developed for neural tissue was developed by Golgi. It's called the Golgi stain, and it had this very, very interesting property. When you stain brain tissue with the Golgi stain, what happens is that a small subset of the neurons become completely filled with a dark black substance called silver nitrate. And when you stain a tissue with this method and you look at the results under a microscope, what you would see is something like this. And as you can imagine, something like this was absolutely game-changing because what it does is it allows you to visualize a neuron in every last little bit of fine structural detail that it has. And being able to do this was really sort of the first major discovery that enabled scientists to understand what the structure of neurons was and to begin to understand their function. And in fact, this observation, this ability to visualize neurons and to visualize their structure and shape led to the development of what's called the neuron doctrine. And it's simply the idea that individual neurons are the fundamental functional unit of the nervous system. Prior to that, there was really no understanding of what the cells of the brain really looked like and how they were shaped or how they were organized. But after the development of the Golgi stain, we were finally able to see what neurons looked like. And this was absolutely revolutionary. So, here, for example, is an illustration of neurons in the human retina. And this is an illustration based upon a Golgi stain. And we can see that within the retina, there's this enormous variety of neurons that have different structures and presumably also have different functions. Right, and so this sort of enormous variety of neuronal structures was something that was completely inaccessible prior to this. In addition to that, in addition to being able to identify cells based upon their structure, this Golgi stain also gives us a sense of how different parts of the brain are organized or how they might be connected. So what we're looking at here is a coronal section through a part of the brain called the hippocampus. So this is actually an illustration that was done by Golgi himself. And what we can see in this illustration is that the cells within the hippocampus have different organization depending on which part of the hippocampus you're looking at. You can see here the cells seem to be very loosely organized, but then over in this section here, they're very clearly organized into a distinct layer. And then as we continue around, we can see that the cells begin to change their shape and the layering becomes less distinct. Right, so being able to visualize neurons in this way gave us huge insights into the structure and function of the brain, just insights that were completely unavailable before then. So really, everything that we're going to be talking about today was stuff that we only began to be able to learn after the development of this sort of revolutionary technique. All right, and here we are now. Here's our illustration of our typical neuron, and I'm just going to go through with you some of the parts that we can see. So like all cells, neurons have a cell body, and within the cell body, we can identify the nucleus, and we can also identify this very important part here, which is this junction between the axon and the cell body. Now this part goes by various names. Here it's called the axon hillock. You can also call it the axon initial segment. This is a very important part of a neuron because this is the area that is primarily responsible for generating the action potential. We're going to talk about action potentials in the next lecture, but action potentials are essentially the signals that neurons send from one to another. Action potentials are generated in the axon hillock and they travel down the axon to the axon terminals. Now we can see here, the next part we're going to talk about is the axon, and you see the axon that's illustrated here isn't very long. In real brains, axons can be very, very long. In fact, if you think of, let's say the neurons that allow you to wiggle your toes, right? So the cell bodies of those neurons are living in your spinal cord, and their axons travel out of your spinal cord and go all the way down your leg, down to the ends of your toes. So those are very, very, very long axons that are sending those signals down to the muscles in your toes that allow you to move them. So they can be very, very long. Axons don't always have just a single target. You can see that they can produce what are called axon collaterals. They can branch off and go to different targets. And in fact, neurons can even send axon collaterals back onto themselves. And a single neuron can send collaterals to many, many different neurons at once. Now, if we go to the ends of the axons, we see what are called axon terminals, or they're sometimes called terminal buttons or terminal boutons, or here they call them end feet or an end foot. What that simply means, and you can see a sort of zoomed in version of a terminal bouton here, what that simply means is that this is the very, very end of the axon. This is, the final destination for the action potential, and it contains a lot of machinery that is necessary for synaptic transmission, which we'll talk about in the fourth lecture. Now up here on the other end of this cell, we see the dendrites, and dendrites can form these very sort of broad, sort of tree-like branching patterns. And the function of the dendrites is to receive synaptic inputs from other neurons. And we can see there's a little zoomed in version of this here. We see this axon terminal here is, forming a synapse with this dendrite right here. And in some neurons, the dendrites even can have what are called dendritic spines. And you can see a zoomed in version here, and you can see it illustrated here. And if you look really close on this diagram, you can even see little tiny spines on the dendrites of this neuron here. And the spines are just these little hairy projections that receive synaptic input and contain a lot of the machinery for the postsynaptic side of synaptic transmission. So these are the basic parts of the neuron. You should know these cold. This is absolutely basic fundamental information that you should be very, very familiar with. All right, the next thing we're going to cover are the different kinds of neurons. And really, we're not even going to talk about too many different kinds of neurons. We're just going to be talking about the ways that we can categorize neurons, the ways that we can think of organizing neurons into types. And there are sort of two main ways of doing this. The first way of doing this is by looking at their structure, looking at their appearance. And this, as I mentioned before, was what the Golgi stain was so good at. What you're looking at here are illustrations of three different kinds of neurons. I think these are probably illustrations that were made based upon a Golgi stain. And you can see, just by looking at these three different neurons, you can imagine that they're going to have, three different functions, that they're going to have different properties from one another. For example, the neuron that's all the way over here on the right, this is its axon here, and this is its dendritic tree. This kind of cell is called a Purkinje cell, which is found in the cerebellum. And just by looking at the dendritic tree of this neuron, you might be able to surmise that an important part of this neuron's function is integrating synaptic inputs from many, many, many different neurons that are going to be synapsing inside this dendritic tree. Over here is another important kind of cell. This kind of cell is very often found in the cerebral cortex, in the neocortex. This is called a pyramidal cell. It's called pyramidal because some people think that when they look at the shape of the cell body here, it looks like a little bit like a four-sided pyramid. And again, you can see the structure here is very, very different. It has a dendritic tree, but it's much, much smaller than this Purkinje cell. Over here, we have what's called a stellate cell. This is a cell that's often found in parts of the hypothalamus. And the name here, stellate cell, comes from the Latin word for star, referring to its sort of somewhat star-shaped appearance. And the one thing about stellate cells is that the dendritic trees, even though they have lots of branches, they tend to be very, very small. So we can presume, from the shape of it there, from its structure there, that it's going to be integrating synaptic inputs, but it's going to be needing to only integrate them over very short distances rather than the very large distances that we see in the larger dendritic trees from Purkinje cells and pyramidal cells. Now, identifying the structures of different neurons is important, but we shouldn't get too carried away with ourselves. The structure here only gives us one clue into their function. And it is very, very possible for neurons that have similar structures to have different functional properties. And likewise, it's possible for neurons that have different functional, different structures to have similar functional properties. So what I mean by that is that there isn't always a one-to-one relationship between a neuron's structure and a neuron's function. Nonetheless, being able to look at these neurons and look at their structure does give us some important hints to what it does. Now, another way of classifying neurons is by their function. And there are two kinds, in general, we can sort of divide neuronal functions into three very broad categories. The first category is sensory neurons. So, if we imagine, if we imagine our toes at the end of our feet, we're able to sense pressure, we're able to sense vibration, we're able to sense heat and cold. And so what that means is there have to be neurons that are, that are detecting those sensations in our, in that part of our body, and are sending that information back into the spinal cord, and then transmitting that up into the brain. So there are neurons whose job it is to detect, sensory features of the world. Likewise, we also have motor neurons. These are the neurons, and I've mentioned these before, that are responsible for controlling our muscles and generating our movement. In addition, motor neurons can also stimulate internal organs and stimulate other bodily functions as well. And I've actually given you before, sort of a clear example of sensory and motor neurons, because we talked about this when we talked about the spinal cord, right? There's a sensory neuron here that's detecting stretch within the quadricep muscles, and likewise, there are motor neurons that are producing contractions within that muscle as part of a reflex arc. Now, if a neuron is not a sensory neuron, and if it's not a motor neuron, then it's going to be an interneuron. And an interneuron simply means neurons that are in between, that aren't sensory or motor neurons. And their function is to connect one neuron to another. And in fact, if we look at our own brain and nervous system, and if we look at the brains of most mammals, most of the neurons in our brain and spinal cord are interneurons, meaning that they don't have a primary sensory function, they don't have a primary motor function. Their job is to connect one neuron to another. And as you could imagine, especially as we get up out of the spinal cord into the brain, these form, sort of vast, vast interconnected networks of neurons. There's this sort of illustration here in the book, and you can see here there's a neuron that is, synapsing on another neuron down here and is receiving inputs from another neuron up there. And it almost gives the impression that neurons within the central nervous system are connected in this sort of serial one-to-one fashion. But this is not true. In real brains, neurons are densely interconnected with one another, and they form very, very complex networks. So this is just a very this is just an illustration of that. So what you're looking at here, this is a figure from a paper that was published in a journal called PLOS Biology. And what it's showing here are neurons within the nervous system of a worm called C. elegans. And this is a worm that's used as a model system in biology and in neuroscience as well. And one of the things about this worm is that it has a very, very simple nervous system. It only has, I think, maybe a couple hundred neurons, which, compared to humans, which have 100 billion neurons, is very, very, very small and very, very simple. And because it has so few neurons, it's possible to generate kind of a wiring diagram to see how they're connected. And what this diagram here is showing is sort of the wiring and the connections between a subset of the neurons in this worm nervous system. And we can identify, for example, neurons that are sensory neurons. And we know they're sensory neurons because they're connected to touch receptors. In addition, we can identify motor neurons, and we know that they're motor neurons because they're connected to the worm's muscles. And what that means is these neurons here are interneurons. They're not sensory and they're not motor, but they help connect the sensory to the motor through direct and indirect connections. And one thing that I hope you can appreciate is that they're not just connected in serial one-to-one fashion, one after another, after another, after another. They have this sort of complex circuit. And one of sort of the great challenges in modern neuroscience is sort of understanding the structure of these circuits, understanding how individual neurons connect to one another and the nature of those connections, and understanding what the function of those connections are, how they enable our abilities for sensation, for movement, for cognition, and so forth. So I just want to emphasize here that most of the neurons in the brain aren't sensory and they aren't motor neurons, they're interneurons, and interneurons form these very complex neuronal networks. All right, now just one very quick aside on this topic of neural networks. You may have heard this term, neural networks, used in the context of computer science or artificial intelligence. And in computer science and in AI, neural networks is a shorthand term, and it's for artificial, and it's a term for artificial networks that are very loosely inspired by neurons and neural architecture, but it doesn't refer to biological networks. Right, so what neural networks in computer science are basically tools that can be used to carry out computationally difficult tasks. For example, on your phone, you probably have some voice recognition capabilities. Those capabilities are enabled by a artificial neural network. And in addition, these networks can be used in some cases for theoretical research on brain function. But when people are talking about Google's latest neural network technology, they're not talking about biological networks. They're talking about artificial networks that are very loosely inspired by neurons. Okay, so we've talked about the basics of neuronal structure, and we've talked about different kinds of neurons, how to think about different kinds of neurons. The last thing that we're going to talk about today are glia. And glia are another class of cells that can be found in the brain, but they aren't neurons, and they don't carry out the same functions as neurons. We can really think of them as being the support system for neurons and for the rest of the brain. The word glia actually comes from the Latin word for glue, and sort of one of the very first and most important functions of glia is providing physical structure to the brain, just kind of holding everything together, holding the neurons in place. In addition to that, glia help provide nutrition and oxygen to neurons. They help form what's called the blood-brain barrier, which I'll talk about in a few moments. And they also have some functions for healing and repair of neurons when the brain is damaged. That's sort of the main primary function of glia. In addition to that, glia also have the important function of producing myelin. And we can think of myelin basically as being a kind of insulation for axons, for the axons of neurons. And this insulation basically helps neural transmission be more energy efficient and be faster inside the brain. And the final function of glia is that they produce cerebrospinal fluid. So you'll remember that your ventricles inside the brain are filled with fluid. The fluid that's in there is called cerebrospinal fluid. And it doesn't just fill the ventricles, it actually surrounds the outside of the brain as well, which provides cushioning and buoyancy to the brain. And cerebrospinal fluid is secreted by a certain class of glial cells. Now, a fun fact about glia is that if we just counted up the number of glial cells and compared it to the number of neurons in the brain, we'd see that glia outnumber neurons by about 10 to 1. You know, you sort of, while we're on this topic, it brings up this old idea that you might have heard that we only use about 10% of our brain. And I think it might come from the fact that only 10% of the neurons in our brain are neurons. The other are glia. But this whole thing about using 10% of our brain is actually totally a myth. It's totally bogus. If I were to put you inside of a brain scanner that monitors the metabolic activity of your brain, what you would see is that almost all of your brain is active almost all the time, even when you're at rest or even when you're asleep. So really, we're pretty much using almost all of our brain all of the time. And the idea that we only use 10% of our brains is just totally made up. Okay, so let's get back to glia. And to sort of summarize the functions of the glia and the different kinds of glia that perform those functions, I actually think this table from the book is really, really good and really summarizes everything very, very neatly. Remember I said one of the functions of the glia is to secrete and to produce the cerebrospinal fluid. That's done by these cells called ependymal cells, and these glial cells live on the walls of the ventricle. And secrete the cerebral spinal fluid directly into the ventricles. You'll remember I talked about another function of the brain, another function of glia being creating what's called the blood-brain barrier. The cells that are responsible for that are these cells called astrocytes, and they're called astrocytes because they have this star shape. And in addition to producing the blood, to supporting the blood-brain barrier, astrocytes also help with healing and brain repair after injury. Another class of glial cells is called microglial cells. These are actually derived from blood tissue, from embryonic blood cells, and their main function is sort of cleaning up and removing dead tissue after injury. And finally, for producing myelin, remember myelin is this sort of insulation for the axons that enhances the efficiency of neural transmission. Myelin in the central nervous system, so in the brain and spinal cord, is made by these glial cells called oligodendrocytes or oligodendroglial cells. And in the peripheral nervous system, so these are the nerves that are running through your body, it's Schwann cells that are responsible for producing myelin. So again, to summarize what glia do, I think this little table just kind of absolutely nails it. It's very, very simple. It's very straightforward. Now, the last thing we're going to talk about in detail is this idea of the blood-brain barrier. And, when I first heard of the blood-brain barrier, I thought that it was something like a wall or maybe like a layer of saran wrap or something that separated off the blood from the brain. I didn't quite know what it was. So I just want to talk a little bit in detail about what the blood-brain barrier actually is. So, so your brain, like every other part of the body, needs blood, it needs oxygen, it needs nutrients from the blood, and it gets those from the capillaries, which are these tiny, tiny, tiny little blood vessels that are, sort of the very end of your circulatory system. And in most other tissues of the body, the capillaries, they're formed by these endothelial cells here, and the gaps and the junctions between these endothelial cells that form these capillaries in most tissues of the body are a little bit loose, and they're a little bit loose so that oxygen and nutrients and other substances can get from the blood into the tissues or from the tissues back into blood. However, if we look at the capillaries that live inside the brain, and we look at the endothelial cells that form those brain capillaries, we would see that the gaps between them are not very loose. In fact, they're sealed up by what are called tight junctions. And what these tight junctions do is they make it a lot harder for stuff to get out of the capillaries into the brain. And what you can think of this as being a sort of form of protection for the brain. Any substance that could potentially harm neurons, is something that could be very, very, very bad and very devastating. So what these tight junctions do is sort of form a kind of protection that protects neurons from bad stuff that might be in the blood. And one of the most important parts of the blood-brain barrier are these tight junctions that form between the endothelial cells. Now, the astrocytes also have a role in the blood-brain barrier. If we were to take, a cross-section here through the capillaries, what we'd see is that the astrocytes would send these projections, these sort of little feet, onto the endothelial cells and onto the capillary, and this would be a means for nutrients and other substances to get from the blood into the astrocyte, and then from the astrocytes into the neurons. So the astrocytes almost sort of serve as kind of a gatekeeping function to make sure that only the substances that should be getting into the neurons actually can get into the neurons. And in addition to that, this whole sort of blood-brain barrier is also supported by these other cells called pericytes, Pericyte, pericytes. So this hopefully gives you an idea of what the blood-brain barrier is, and this is something that's actually very, very important in medical contexts. So for example, there are many, many drugs that if you take them will make its way into most of the tissues of the body, but won't make its way into the brain because they can't cross the blood-brain barrier. So if you're going to be studying any kind of internal medicine or pharmacology, it's very important for you to understand which drugs can cross the blood-brain barrier and which drugs can't. All right. So we've come to the end of our lecture, and we're going to do, a little quick test of your knowledge. And the most basic thing I can think of here is if I just kind of gave you this sort of simple unlabeled picture of a neuron, I'd ask you how many of the different parts that you can see and that you can label spontaneously. Now I can think of 7 that you should be able to label off the top of your head. So if a moment ago you could only think of 2 or 3, I'd say go back and study the slide where we talked about the structure of the neuron. If you could do 5 or 6, that's great. And if you could do all 7, that's really excellent. Now that I've prompted you, now that you can see the parts that I've highlighted, hopefully you should be able to name all of these. I'll give you a moment just to sort of mentally go over them and see if you can. And here are all of the parts that I think you should just be able to label right off the top of your head. And again, if this was difficult for you, go back and review the slide where we went in depth and in detail. Go read the book and get yourself caught up on that. All right, here's a summary of what we talked about today. In the next lecture, what we're going to be doing is we're going to be basically getting ourselves ready to talk about one of the most important functions of the neurons, which is the resting potential and the action potential. All right, catch you next time.